Leukocytes, or white blood cells, normally circulate in the blood unattached. The presence of an infection or inflammation triggers leukocytes to move into tissue toward the foreign invader in a process called extravasation. Extravasation requires a succession of specific cell-cell contacts between leukocytes and endothelial cells lining the blood vessel. First, inflammatory signals induce endothelial cells to exocytose P-selectin and synthesize platelet activating factor, PAF, both of which appear on the cell surface. P-selectin molecules bind carbohydrate groups at the leukocyte cell surface. This weak binding to selectin, coupled with the force exerted by the flowing blood, causes the leukocytes to roll. The leukocyte also contains PAF receptors and integrins. Receptor binding to PAF results in the activation of the integrins. The activated integrins are able to bind ICMR molecules constitutively expressed on the surface of endothelial cells. This causes the leukocyte to stop rolling and to tightly adhere to the endothelium. The adhered leukocyte then begins its migration between adjacent endothelial cells to reach the underlying tissue.